Okay, hello everybody. So my research this summer and in the school year and beyond this summer is focused on the generation of limbal stem cells um, to offer a treatment, an improved treatment for patients that have limbal stem cell disease, which causes roughly 15 to 20% of corneal blindness worldwide. Limbal stem cells are an ocular stem cell type, and they are responsible for the maintenance of the cornea. So that is the clear covering of your eye over the iris and the pupil. They reside in the limbus. So the limbus is next to your iris, and it forms the black ring around your iris that divides, of course, the colored part and the white part of your eye. So they reside in the limbus. And when the cornea is insulted, so through a scratch or what have you, or through normal cell shedding, desquamation, the limbal stem cells begin to divide asymmetrically. So they create a daughter stem cell and then a non-stem cell. And this non-stem cell migrates from the limbus circumferentially to the center of the cornea. And along that path, it differentiates into a transit amplifying cell and then a corneal epithelial cell finally, all expressing different genetic markers. So as I mentioned, the point of this research is for clinical treatments for limbal stem cell deficiency. And the reason why we are doing this is because current treatments, really the most effective current treatment is donor transplants, which are not successful in the long term because, well, it's donor tissue and typically people's immune system rejects the transplants after a while. And so what makes our research special is that we're using induced pluripotent stem cells. So induced pluripotent stem cells, of course, are stem cells that have had their pluripotency induced. So they come from an adult cell in your body and they are reprogrammed into a stem cell. And so that would bypass any need for a donor in the case of limbal stem cells and in regenerative medicine as a whole. And so people could, with limbal stem cell deficiency, could function as their own donors, not have to worry about any tissue rejection and live life. And so my project began last summer, and we started following a protocol from a Finnish group that proposed to generate limbal stem cells in about 11 days through a four-day ectoderm induction, and then plating these cells that have been committed to ectoderm onto a matrix of collagen 4 and laminin 521, and then differentiated in a corneal epithelium medium. So I did that, but upon searching for genetic markers, we noticed that a putative limbal stem cell marker, ABCG2, which is thought to be a stem state marker, disappeared after about 1 week in adherent culture on the collagen 4 and laminin 521. And so that was an issue, but about 1 month later, the same group published a follow-up protocol that proposed to maintain this expression, the expression of ABCG2, which would maintain the stem state and would allow for perhaps functional transplants. These limbal stem cells would be able to have regenerative potential instead of just being passaged onto something and dying off. So that is what this research is about. So as I mentioned, I followed a four-day ectoderm induction in suspension. And then I plated these cells down onto the collagen laminin matrix. And for 1 week, I used a corneal epithelium medium to commit them to become some kind of corneal cell. And then after 1 week in culture, I used a cocktail of factors that have been implicated, at least in other stem cell populations, to maintain their stem characteristic. So the stem population in question is the intestinal stem cell niche. I'm sorry, the intestinal stem cells. And these factors maintain the intestinal stem cells through the modulation of the Wnt pathway and the BMP pathway. So the BMP pathway gets inhibited and the Wnt pathway gets activated, and the intestinal stem cells continue to express ABCD2, like a limbal stem cell expresses ABCD2, and they maintain the regenerative potential. So that is what the protocol proposed. And upon replication, or at least partial replication, we found similar results. So after day 11, after the peak expression of ABCD2 was noted in the previous protocol, we got to day 18 and we noticed that ABCD2 was still pretty strongly expressed. And so that is good news. But we also noticed that we couldn't really do a lot with the cells. We were not able to lift the cells off of the plates that they're on. They would die or they would turn into something else. They would differentiate. And so that's a major issue. Also, these conditions, these culture conditions, these new culture conditions are very harsh to the cells. So I will show you the phase images of these cells in the supplemental material, and you can see that they get selected against pretty harshly. And it's unclear why that is. Either the conditions are selecting for the limbal stem cell type, or they're just selecting for anything that may express ABCD2, but may not be a limbal stem cell. We don't know. And then another thing is neither the publishers of the protocol that I followed, nor us, stand for CG2, or I'm sorry, ABCB5, another putative marker of the limbal stem cell type, post day 11. So it's whether or not that marker is maintained. And if that's not maintained, then the cultures will not function as a transplant. And also, in some communication with the author of this protocol, which was very cool, she mentioned that there is cell line specific results, which means that this protocol is going to have to be optimized to the point where there would be no cell line specific results. It would have to work in all cases, or otherwise with every patient, if it ever got to clinical practice, every patient would have to have an optimized protocol, which would be weird. So just with our findings, so we got that ABCD2 is maintained, at least to day 18. But going forward, we're going to need to compare, so the donor cells to these iPSC generated limbal stem cells. We're gonna need to be able to do functional comparisons, further phenotypic comparisons, so that we're very sure that we're generating what we want to be generating, because there is no universally accepted marker of the limbal stem cell type. And so we rely on a multitude of markers to indicate the stem cell type. So yes, here are some up close pictures so you can see what's going on. So here is the day 11 time point that I mentioned before, that in our old protocol yielded us the highest expression of ABCG2. And this is what it looks like at day 11. Just looking for a nuclear marker P63, which we won't worry about, but you can see that ABCG2 is membrane bound, it's highly expressed, or at least where it is expressed, it's strongly expressed, and that's in red. But then you can notice underneath, so CNT 30, day 18, this is following the old protocol post day 11, so post that peak expression. And you can notice that there is no red in the culture. So that means that there's no ABCG2. But if you go down to the bottom here, in our new culture conditions at day 18, you can see that there is ABCG2 maintained. You'll note that instead of dual expression between P63 and ABCG2, you kind of get two camps of cells where you would get ABCG2 expressed largely on its own, and then P63 expressed in green around the ABCG2. So that's promising. And then these are a little difficult to see. They're not the best pictures. I did not take them, so please don't shoot the messenger. But these, so on the right, I mentioned ABCB5 before. This marker specifically was knocked out in mice. And when it was knocked out in mice, the knockouts were observed to have developmental abnormalities in their cornea and in their limbus. And upon injuring the cornea, and upon histological analysis, the cells that regenerated the cornea did not epithelialize correctly. And so ABCB5 is thought to be the most specific marker of the limbal stem cell type. But again, there would still need to be a consensus. We still rely on it as a putative marker. So this is day 11. So this is 1 week in culture. This is our peak expression time point in our old protocol. And you can see that ABCB5, which is in red, is not really expressed. It's a membrane protein like ABCG2 is, but it's not highly expressed. You can see it's expressed a little over here, but in general, it's still nucleus bound. It's still in the nucleus along with P63, which is why you see this kind of orangish weird color. Whereas when you look at ABCG2 at 1 week, or at day 11, you can see that it is in the membrane, given all of the red, and that P63 is being expressed in the nucleus without ABCG2. So if ABCB5 is truly, if ABCB5 is LSC specific, one thing that we're going to need to do is we're going to need to be able to find the point at which both of these markers are expressed, ABCG2 and ABCB5, or populations where they're both expressed, and purify them. And that is what the further research that that my lab is going to be doing is going to be focused on, purifying these cells, doing functional assays, and all of that jazz. And then here I mentioned harsh selective conditions imposed upon these cells by the new ENRC conditions. And so you can note that after 1 day in culture, so on the far left, day 12, that's 1 day post adding this cocktail of factors. And you can see the cultures are largely unaffected, but by day 2 and day 3, you can start seeing things are starting to die off. And then you get all the way down to 1 week, day 18 over here, and you don't really have that much left. And it's not clear what's going on. I have some hypotheses, but that's something to watch out for too, because we don't know what's getting selected against.
Hey, Isaias. I'm gonna stop you there if that's okay, and open it up for questions, because I want to make sure that people who have questions have a chance to answer or ask them, and you can answer them. So does anyone have any questions for Isaiah?
Anybody?
I have a question.
Yep.
When you talk about, it was just at the end, so in your supplemental materials, you talked about having a few hypotheses for what could be selected against. Could you elaborate on that?
Yeah, so ABCG2, that thing that's being like hailed as a stem state marker. It's found in stem cells, but it's also found in cancer cells too. And so its other name is the breast cancer resistance protein, I believe, breast cancer resistance gene. And so it was discovered because when a population of cancer cells was being treated, this membrane transporter was transporting the anti-cancer drugs out of the cells and protecting and allowing the cells to live. So one of the small molecules in the cocktail factor, Chier 99021, the C in this acronym, it's a Wnt activator, but what it's suspended in, what it's reconstituted in is DMSO, dimethyl sulfoxide, which is toxic to cells at a certain concentration. I don't know what the concentration of DMSO is in this culture, in the cultures, but if it is toxic, what could be happening is that, now my mentor is here, so if he thinks this is wrong, please tell me, James. But what could be happening is perhaps, I don't know, the DMSO, the Chier, at least the DMSO is getting transported out of the cell. I don't know if it works like that, but it's just weird to me, because we've seen the ABCG2 is surviving, and so, but not so much the P63 positive cells. These are not surviving at all, really. I mean, there's a lot fewer, and so maybe the ABCG2 is conferring some benefit, or I'm really not sure how these cells are being selected for and against. Sorry, that was really convoluted, but thank you, Michelle.
I had a question, Isaias.
Yeah.
I missed the origin of the starting cells you used.
They're iPSCs, so they, the starting cells we used are down here. They're these, so they're iPSCs. They are induced pluripotent cells. They're on a laminin matrix.
Okay.
And then from their adherent culture as stem cells, we suspend them, we create embryonic bodies, we induce ectoderm, and then we plate them down onto the collagen laminin matrix.
Mm-hmm. And they need to stay in the matrix. Once they come off, they start to die. Is that what you observed?
I've not had successful passages yet. Yeah, they either die or they differentiate.
Uh-huh.
Yeah.
And in theory, you could take cells from each patient and go through this process and regenerate the.
Yeah, but ideally the protocol would be optimized so that we wouldn't have to. So I mentioned that this seems to be cell line specific, and that's confirmed by the author. She mentioned that. So ideally, the protocol would be optimized to the point where we don't have to individually optimize the conditions for each patient. Yeah, but yes, each patient would be able to be their own source of limbal stem cells.
Yeah, that sounds terrific.
Yeah.
Thank you.
Cool. Anyone else?
Any other questions for Isaiah? Nice job, Isaiah. Thank you. I believe Heidi is making you a presenter, Kenan. If you want to get your materials up, we'll wait 2 minutes to start. So in case you have people coming, they will be there.
Sure thing.
So I had another question for Isaias while we're waiting.
Absolutely.
What's needed to induce like a pluripotent stem cell to go down this lineage? What kinds of treatments did you need to do?
We used, So we used, so the embryonic body formation is one thing. So when the cells aren't attached to anything, they're not going to be communicating, they're not going to be signaled to by the matrix. So that's one thing. And that allows for us to knock out germ layers through, we use fibroblast growth factor, basic fibroblast growth factor, and a small molecule SB550124. That knocks out mesoderm.
Okay.
And I believe endoderm. I know mesoderm for sure. And then after that, we use bone morphogenetic protein 4 to induce ectoderm. Yep.
Great.